Hi everyone, welcome to this week's Science Podcast and happy Friday the 13th. I am thrilled to tell you about a new publication in the Journal of Autism and Developmental Disorders around the different names for early behavioral interventions for autism. But instead of listening to me talk, Dr. Laura Schriebman, director of the UCSD Early Intervention Research Program and world-renowned expert in early behavior intervention, is going to tell you why this study is important for clinicians, parents, educators, legislators, and insurance companies.
The idea for this paper came about in 2012 at the Autism Speaks Toddler Treatment Network meeting at the International Meeting for Autism Research. And at that meeting, several of us researchers, both with behavioral orientation and developmental orientation, got together and decided that there really needed to be some clarity about some of the early interventions that were proving to be so effective for these children. And naturalistic developmental behavioral interventions have been around for a long time, and they've gone by names like pivotal response training, early start Denver model, incidental teaching, and others. And these interventions are really representative of the merging of both applied behavior analysis and developmental science. And with our ability to diagnose these children earlier and earlier, and the importance of early intervention was really a strong motivator for us to get out to the field for some clarity in the different interventions. Since these interventions have different names, and in fact they do have some differences between them, the fact is that they are empirically validated and they share many common features. They're conducted in natural settings, they're manualized, they have fidelity of implementation measures, individualized treatment goals. So because of these commonalities across these different interventions, it became pretty clear to us that people needed to see that there is a general approach of naturalistic developmental behavioral intervention. And we thought this was necessary to show potential consumers like parents and teachers and clinicians and so on that, these interventions that may have different names really do adhere to the general idea of a naturalistic developmental behavioral intervention. We also thought it would be important for insurance companies and other funding agencies to appreciate this, the fact that these are at least as empirically supported, if not more so than what they call ABA interventions, which we would call discrete trial interventions, which are more highly structured. So that these interventions, the NDBIs, they're conducted in naturalistic settings, and they involve much more child involvement. And we wanted to get that out there, that these different interventions in the field should now identify themselves as a form of NDBI, so that there won't be a problem of people saying, are you getting pivotal response training or incidental teaching, when in fact it probably would be the same in terms of outcome.
What's even most remarkable about this paper is that it really brought together the authors of these different types of interventions, the ones that were actually writing the manuals and developing the protocols for things like early social interaction and early start Denver model. They all agreed that researchers now need to start using the words naturalistic developmental behavioral intervention. The consistent use of this term will help policymakers, families, researchers, physicians, and other treatment providers understand where a specific intervention exists within a continuum of intervention practices. They also recognize that future research should lead to interventions that are even more effective, efficient, and individualized. Better understanding of the active ingredients of the interventions, how well they can be turned into protocols that lead to good outcomes in community settings, and the components that have the strongest effect on outcomes for subgroups of children with different symptoms should really be emphasized. On the same topic of early detection and early intervention in autism, a new study out of the University of California at San Diego has reported early data on a new biomarker for autism that's aimed to help make a diagnosis in toddlers. There's actually many of these types of blood-based genetic analyses under research, but what makes this one different is that they're not just looking at mutations of a few genes. Rather, they narrowed down a long list by those that had more complex interactions and those that are shown to be differentially expressed in brain tissue. This concept is actually easier to look at on a figure than it is for me to explain it to you, so I got permission from the authors to post one of the key figures for you to see. The authors analyzed blood from toddlers with autism and those without autism and compared which genes were expressed differently. Some were expressed at higher levels, some were expressed at lower levels. Then they ran the study again in a new group, validating the findings of the first study. They first identified over 2,700 genes that were differentially expressed and then organized them based on what they did. For the most part, they're involved in turning genes into proteins, then inflammation, cell adhesion, and the immune response. This is interesting given that the immune response has been studied as a moderator of autism symptoms, and a recent study of brain tissue also identified an immune cell as a key participant in genetic signaling. Then they used databases of what all of these 2,700 genes do to describe them in terms of how many networks they formed. If they had more than 10 to 20 interactions, they were considered the most complex and put in the middle of the graph. The ones at the center, which again is posted next to the podcast on our website and our Facebook page, were also ones that are seen in autism brain tissue. What makes this unique is they were able to provide evidence of overlap with brain tissue findings, strengthening their conclusions. They also looked at kids from general pediatric clinics and those with pediatric concerns. They found an 83% accuracy, meaning they could accurately classify a person with autism 83% of the time. It's important that they use kids from general pediatric clinics and those with concerns. Many times, these reports of biomarker studies are done in samples of kids of what we already know have autism, making their utility somewhat limited because we can't really apply them to individuals that we may suspect have autism. The toddlers in this study showed the sorts of behaviors we would expect to see in community clinics, while the ones without autism are the types you'd see with typical development or some global concerns like is a language delay, transient language delay, or global developmental delay. Because the description of autism is becoming wider and wider, and autism is becoming more and more prevalent, more biologically based descriptions of autism are necessary. And this is a perfect example of how brain tissue is informing biomarker studies and strengthening data behind creating